This is a presentation on the study of the Drosophila melanogaster by Isabella Frullo and Lily Clardy. In this experiment, we studied wild-type flies, single mutant flies of Sapia mutation, and double mutant flies of the Apteris Sapia mutation. There are 3 stages of a fruit fly's life cycle: the egg, the larva, and the pupa. Fruit flies develop to adults in about 1 to 2 weeks, and they last as an egg and larva for about 8 days. The pupa stage lasts about 6 days, and adult flies live for several weeks. The wild-type phenotype of an adult Drosophila melanogaster is about 1/8 of an inch long. Its body is split into 3 sections: the head, the thorax, and the abdomen. Their bodies are a light brown or tan color, whereas an immature fly is more clear. Its eyes are teardrop-shaped, bright red, and about half the size of their heads. There are 2 wings that are clear, elongated, and oval-shaped, attached at the thorax. Female flies have a more pointed abdomen, whereas the male flies have a more rounded abdomen. The most reliant way to tell the difference in sex is to check if there are sex combs, which are dark lines on the front legs of the flies, which is a trait mostly found on males.
When looking at the development of eye color of flies, the fly's eyes are usually large and colored, and they can range anywhere from the color red to sepia to white, and it shows a lot about the fly's genetic makeup when you're looking at the color of their eyes. So fruit flies bred in the wild typically have red eyes, but brown and sepia eyes are more of a result of a recessive gene that occurs when 2 sepia-eyed flies mate. And then white fruit flies are usually less common, and they typically only occur from mutations.
The single mutant flies studied in our experiment were called Sapia. They are physically identical to the wild-type flies. However, instead of having the red eyes that are found on the wild-type flies, Sapias have a dark brown or black-colored eyes. Immature flies have a bit of a lighter color brown, whereas mature flies have almost black eyes. The gene Sapia is 938 megabase pairs and has a cytogenetic location on chromosome 3 on the left arm, found on the positive DNA strand. It has a homologue GSTO1 found in Homo sapiens. The double mutant flies found in this study were called Apteris sapia. They have the same color eyes as the Sapia mutant and have a lack of wings. Where wild-type flies have 2 wings attached at the thorax, Apteris do not. This mutation causes the flies to be 70% more viable than the wild-type flies, but more erratic, having a shorter life cycle. This causes both sexes to be sterile. The protein coded for by the Apteris gene is required for the regular development of the wing and halter imaginal discs. A mutation in this gene will lead to irregularity in the development of these discs, therefore resulting in the inability of the flies to develop wings. The Apteris sapia gene is 21,324 megabase pairs long and is located on chromosome 2 on the right arm, found on the negative DNA strand. It has a homolog LHX found in Homo sapiens. 4 parental crosses were made from this experiment. They were made in 3-to-1 ratios of virgin females to males. The first cross was of wild-type males and single-mutant females. The second cross was of single-mutant males and wild-type females. The third cross was of wild-type males and double-mutant females. And the fourth cross was of double-mutant males and wild-type females. From each parental vial, 10 males and 10 female flies from the F1 generation were crossed. The general steps to this method's portion of the lab were that flies were first anesthetized using FlyNap and were then sexed under the microscope, and the number of males and female flies were taken note of in our lab notebooks. After that, the crosses were made, and adult flies were cleared a week later. Flies from the F1 generation were then sexed and phenotyped, and the F1 crosses were made by taking 10 F1 males and 10 F1 females from each parental cross and putting them into new vials. Two PCR reactions were done in this experiment. The first one was the wild type, and the second one was for the single mutants. The reagents in the PCR were the master mix, primers 1 and 2 for the wild type trial, nuclease-free water, and fly genomic DNA, which was calculated using DNA extraction. For the single mutant, all the reagents stayed the same, but primers 3 and 4 were used instead of primers 1 and 2. Our hypothesized results of the single mutant crosses of the F1 generation were that all of the produced flies would have the wild-type phenotype, which is the red eyes. We found this by making predicted Punnett square for the F1 cross. These were not the results that we found. Instead, for both the single mutant males with the wild-type females and the wild-type males with the single mutant females, both had single mutant genes present, so both crosses showed the sepia eyes. So the crosses were not pure, which could be due to many possible errors in the experiment. For the wild-type flies, fragments were expected to be seen at 200 base pairs, 400 base pairs, and 800 base pairs. For the single mutants, there should have been a fragment at about 0.45 base pairs. In ladder 1 of the single mutant trial, there was the 1 kilobase ladder. In ladder 2 was the wild-type DNA. In ladder 3 was the single mutant DNA. And in ladder 4 was the 100 base pair ladder. There was a fragment shown at about 0.5 kilobase pairs in ladder 2 of the single mutant trial. The wild-type ladders did not show the expected results. In ladder 1, there should have been a fragment shown at 200 base pairs. In ladder 2, there should have been a fragment also shown at 200 base pairs. In ladder 3, there should have been a fragment at 400 base pairs. In ladder 4, there should have been a fragment at 200 base pairs. And in ladder 5, there should have been fragments shown at 200, 400, and 800 base pairs. We originally hypothesized that the flies would follow Mendelian inheritance patterns. However, based on our results from the gel electrophoresis and from the crosses, our data did not support this hypothesis. However, this could be due to possible errors that occurred during our experimental process. For example, flies could have been sexed incorrectly. The flies could have been not properly homogenized during the PCR reaction and the gel electrophoresis, or flies could have mated too soon before we were able to clear the vials, and the crosses were impure. We originally expected the results of the double mutant crosses of the F1 generation to be of 100% wild-type phenotype. However, this was not the case. For the wild-type males and double mutant female cross, the cross was not pure. Instead of having all flies present the wild-type phenotype, flies showed the sepia-colored eye mutation gene, and as well as the Apteris gene. So a lot of the flies didn't have wings, where all of them should have had wings. And in the double mutant male and wild-type female cross, none of the pupa had hatched, and we weren't able to collect any data from that cross. Since none of our crosses had worked successfully, we were not able to retrieve any F2 data because none of our flies had gotten to that point.
So for future work, we would love to continue research with crosses that failed during experimentation or didn't go as planned. And we would also like to explore reproduction rates between different types of mutants to explore quick or slow reproduction rates of certain mutant crosses. Some lessons learned during experimentation are that flies should be checked on more consistently to ensure less error during experimentation and within the crosses, because being non-consistent can lead to death or unwanted crosses. We also learned that when checking on flies, make sure that they have enough food, or else they may die, or like I said before, it could lead to the unwanted crosses or the death.